Close your eyes for a moment and imagine holding a teaspoon of rich, dark garden soil in your hand. The earth feels cool and crumbles slightly between your fingers. It smells of rain and growing things. Now, here's something that might surprise you. In that single teaspoon, smaller than what you'd use to stir your morning coffee, there exists an entire universe of life more populous than all of humanity. Billions upon billions of microscopic organisms live in that tiny space, each one going about its business, invisible to your eyes but vital to the world you know. Welcome to Bedtime Biology, where science meets serenity. Each night, we explore the quiet wonders of the natural world, calming your mind and sparking your curiosity. Please like, subscribe, and comment below with where you're checking in from. Tonight, we're diving into one of the most extraordinary stories in all of biology. A story that challenges everything we thought we knew about who really runs this planet. We live in a world that seems designed for creatures like us, for mammals and birds, for the things we can see and touch. We build cities and reshape landscapes. We pride ourselves on being Earth's dominant species. But the truth is far more humbling and far more fascinating. The real rulers of this world are so small that trillions of them could fit on the head of a pin. They've been here far longer than we have. They outnumber us by numbers so vast they're almost incomprehensible. And without them, not a single one of us could survive even a week. They are bacteria, and this is the story of why they rule the world. Let's begin by understanding just how thoroughly bacteria dominate our planet. When we think about life on Earth, we tend to think about the things we can see with our naked eyes. We think about forests full of towering trees, oceans teeming with fish, savannas roamed by great herds of animals. These are the images that fill our nature documentaries and capture our imagination. But these visible creatures, as magnificent as they are, represent only the tiniest fraction of life on Earth. The real action, the real abundance, the real diversity of life exists in a realm invisible to us, in the microscopic world of bacteria. Consider this remarkable fact. Bacteria are estimated to make up more than 3/4 of all species of life on our planet. Think about that for a moment. Not 3/4 of all microscopic life, but 3/4 of all life, period. Every plant species, every animal species, every fungus, and every other living thing you can name. All of that combined makes up less than 1/4 of life's diversity. The rest belongs to bacteria and their close relatives, the archaea. But it's not just about the number of species. It's about the sheer number of individual organisms. Scientists have calculated that there are approximately 4 to 6 times 10 to the 30th power bacterial cells on Earth. That's a 4 followed by 30 zeros. To put that in perspective, there are only about 10 to the 24th power stars in the observable universe. There are more bacterial cells on Earth than there are stars in 1,000,000 galaxies like ours. And here's something even more mind-bending. These bacterial cells aren't just scattered randomly across the planet. They're everywhere. In the soil beneath your feet, in the water you drink, in the air you breathe. They coat every surface, including every surface of your own body. In fact, the number of bacterial cells living in and on your body right now outnumber the cells that make up your body itself. You are, in a very real sense, more bacterial than human. One particular species of bacteria, with the somewhat unwieldy name of Candidatus pelagibacter ubique, holds the distinction of quite possibly being the most abundant organism on Earth. This tiny bacterium lives in the world's oceans, and there are so many of them that if you could somehow gather them all together, they would weigh more than all the fish in the sea. Scientists have estimated that there may be 2 times 10 to the 28th power individual Pelagibacter cells in Earth's oceans. That's 200 billion bacteria of just this one species living in our seas. To give you a sense of scale, there are approximately 10 million times more individuals of this one species of bacterium in Earth's oceans than there are stars in the entire observable universe. Let that settle in for a moment. One species of bacterium, invisible to the naked eye, exists in greater numbers than the stars themselves. And Pelagibacter is far from unique. The ocean harbours an estimated 10 to the 29th power bacterial cells in total, representing countless thousands of different species. The soil is even more densely packed with bacterial life. A single gram of healthy soil, about the same as that teaspoon we imagined earlier, can contain between 100 million and 1 billion bacterial cells, representing thousands of different species. Some of these bacteria have been living in the soil for millions of years, developing intricate relationships with the plants that grow there and with each other. When we zoom out to look at the global picture, the dominance of bacteria becomes even more apparent. In terms of total biomass, the total weight of all living things on Earth, bacteria make up a staggering portion. While plants dominate in terms of total biomass because of their large size, bacteria punch well above their weight class. Despite their microscopic size, there are so many of them that together they account for approximately 80 gigatons of carbon. That's 80 billion tons. To put that in perspective, that's more than all the animals on Earth combined, including all the fish in the sea, all the birds in the air, all the insects, and yes, all 7 billion humans. But bacteria's rule over Earth isn't just about numbers, it's about time. They've been here longer than any other form of life. Much, much longer. While humans have existed for perhaps 300,000 years, and complex multicellular animals have been around for about 600 million years, bacteria have been evolving and thriving for nearly 4 billion years. They emerged when Earth was still a young planet, when the oceans were hot and the sky was a different colour. They've witnessed the birth and death of continents. They've survived ice ages and asteroid impacts. They've seen the rise and fall of countless other species. And through it all, they've not just survived, they've thrived. This extraordinary longevity has given bacteria time to evolve into forms that can survive in virtually every environment on Earth. From the frozen wastes of Antarctica to the boiling hot springs of Yellowstone, from the deepest trenches of the ocean to miles beneath the Earth's surface, bacteria have found a way to make a home. They can survive levels of radiation that would kill any other organism. They can live in acid so strong it would dissolve metal. They can thrive in salt concentrations that would dehydrate any other cell. They can even survive in the vacuum of space, as experiments on the International Space Station have proven. This remarkable adaptability stems from bacteria's unique evolutionary advantages. Unlike larger, more complex organisms, bacteria reproduce incredibly quickly. Under ideal conditions, a single bacterium can divide into 2 every 20 minutes. That means that in just one day, a single bacterial cell could theoretically give rise to more descendants than there are atoms in the universe. Of course, this doesn't happen in reality because resources are limited, but it demonstrates the incredible reproductive potential of these organisms. This rapid reproduction rate means that evolution can happen at an extraordinary speed in bacterial populations. Mutations that might take thousands or millions of years to spread through a population of larger organisms can sweep through a bacterial population in a matter of hours or days. This is why bacteria can develop resistance to antibiotics so quickly, and why they can adapt to new environments with such remarkable speed. But bacteria don't just rely on random mutations for their evolutionary flexibility. They have another trick up their sleeve, if you'll pardon the metaphor for an organism without sleeves. Bacteria can share genes with each other through a process called horizontal gene transfer. Unlike larger organisms, which can only receive genes from their parents, bacteria can pick up useful genes from their neighbors, even from bacteria of entirely different species. Imagine if you could acquire the ability to breathe underwater simply by spending time with a fish, or develop night vision by hanging out with an owl. That's essentially what bacteria can do. If one bacterium develops a useful trait, say the ability to break down a new food source, or resist a particular antibiotic, it can share the genes for that trait with other bacteria around it. This allows bacterial populations to adapt to new challenges with astonishing speed. This gene-sharing ability has enormous implications for the bacterial world. It means that beneficial adaptations can spread through diverse bacterial communities much faster than they could through simple reproduction. It's like the difference between evolution with and without the internet. Organisms that can only pass genes to their offspring are limited to a slow, generational pace of change. Bacteria, with their ability to share genes horizontally, can spread innovations through their population at revolutionary speed. Now, you might be wondering, if bacteria are so abundant, so ancient, so adaptable, why don't we notice them more? Why does it seem like larger organisms dominate the world? The answer lies in our limited human perspective. We evolved to perceive the world at our scale. We notice things we can see, things that might help us or harm us at a visible level. A lion is relevant to us. A tree is relevant to us. A bacterium at 1/1,000,000th of a meter across is simply too small for our brains to have evolved mechanisms to detect directly. But just because we can't see bacteria doesn't mean they aren't having an enormous impact on our lives and on the planet as a whole. In fact, as we'll explore throughout this journey, bacteria are so integral to the functioning of Earth's systems that without them, the planet as we know it would cease to exist. The air we breathe, the soil that grows our food, the cycles that distribute nutrients through the environment, all of these depend utterly and completely on bacteria. Let's travel back in time now, back to an Earth that would seem utterly alien to us. Picture a planet approximately 3.8 billion years ago. The oceans are warm, almost hot. The continents, if you can call them that, are small and scattered. But the most striking difference is the sky. Look up and you would see no blue. The atmosphere is thick with methane, ammonia, and carbon dioxide. There is no oxygen, or at least not free oxygen in any significant amount. The sky might appear orange or reddish, painted by the haze of methane in the atmosphere. In these ancient oceans, in conditions that would seem hostile to any life we know today, the first bacterial cells emerged. We don't know exactly how life began. It remains one of the great mysteries of science. But we do know that once it appeared, it took the form of simple, single-celled organisms, remarkably similar to the bacteria living today. These ancient bacteria were anaerobic, meaning they lived without oxygen, because oxygen simply wasn't available. For more than 1 billion years, bacteria were the only form of life on Earth. They evolved slowly, diversifying into different forms, developing new ways to harvest energy from their environment. Some learned to use chemicals like hydrogen sulfide or iron. Others developed the ability to harvest energy from sunlight, though not in the way modern plants do. These early photosynthetic bacteria used simpler molecules and didn't produce oxygen as a byproduct. Then, somewhere around 2.7 billion years ago, something revolutionary happened. A group of bacteria evolved a new form of photosynthesis, one that used water as a source of electrons and produced oxygen as a waste product. These bacteria were the ancestors of modern cyanobacteria, sometimes called blue-green algae, though they're not algae at all. At first, the oxygen produced by these cyanobacteria didn't accumulate in the atmosphere. The early Earth had enormous reserves of reduced minerals, particularly dissolved iron in the oceans, and volcanic gases rich in hydrogen and methane. The oxygen produced by cyanobacteria reacted immediately with these substances, oxidizing them. The iron in the oceans rusted, forming distinctive red layers in rocks that we can still see today. These banded iron formations are beautiful evidence of this ancient chemical battle between biology and geology. For hundreds of millions of years, cyanobacteria pumped oxygen into the environment, only to have it consumed by these chemical reactions. But gradually, inexorably, the balance began to shift. As the Earth's supply of easily oxidized materials was used up, oxygen began to accumulate. Not quickly, not all at once, but steadily, the composition of Earth's atmosphere began to change. Then, approximately 2.4 billion years ago, something dramatic happened. The concentration of oxygen in the atmosphere suddenly jumped. This event, known as the Great Oxidation Event, was one of the most important moments in Earth's history. For the first time, the planet had significant amounts of free oxygen in its atmosphere. The consequences were enormous. For many of the anaerobic organisms that had dominated Earth for over 1 billion years, oxygen was a deadly poison. It was a disaster, a mass extinction event that wiped out countless species. It's been called the oxygen catastrophe, and that's probably an appropriate name from the perspective of those oxygen-intolerant bacteria. In a matter of geologic moments, bacteria had transformed the entire planet, and many of their fellow organisms paid the ultimate price. But the Great Oxidation Event wasn't just a catastrophe, it was also an opportunity. Oxygen is a powerful chemical, and organisms that could tolerate it, or better yet, use it, had access to an entirely new and much more efficient way of generating energy. Aerobic respiration, the process of using oxygen to break down sugars and other organic molecules, yields far more energy than any anaerobic process. Bacteria that evolved the ability to use oxygen had a huge advantage. More importantly, for the long-term history of life, the oxygenation of the atmosphere paved the way for the evolution of all complex life. Every animal that has ever lived, from the smallest insect to the largest whale, depends on oxygen for survival. Without the Great Oxidation Event, without those ancient cyanobacteria pumping oxygen into the atmosphere for hundreds of millions of years, none of us would exist. But the cyanobacteria weren't finished shaping the world. The oxygen they produced had another critical effect. It led to the formation of the ozone layer. High in the atmosphere, ultraviolet radiation from the sun split oxygen molecules into individual oxygen atoms. These atoms then combined with other oxygen molecules to form ozone, O3 instead of O2. This ozone layer absorbed harmful ultraviolet radiation, making the Earth's surface much more hospitable to life. Before the ozone layer existed, the surface of Earth was constantly bathed in intense ultraviolet radiation that would quickly damage DNA and kill cells. Life could only exist in water, which provided some protection, or in other sheltered environments. But with an ozone layer in place, life could safely colonize the land. Every terrestrial organism, every plant and animal that lives on land today, owes its existence to the ozone layer, which in turn owes its existence to bacterial photosynthesis. Think about that for a moment. Bacteria, these microscopic, single-celled organisms, fundamentally transformed the entire planet. They changed the composition of the atmosphere. They made the surface habitable. They made possible the existence of all complex life. And they did all of this not through conscious design, but simply by going about their business, by photosynthesizing and reproducing, by doing what bacteria do. This is one of the most profound lessons of biology. Small things, given enough time and enough numbers, can reshape the world. The mighty cyanobacteria, each one invisible to the naked eye, each one concerned only with its own survival and reproduction, collectively wielded the power to transform a planet. They still do. Modern cyanobacteria continue to play a vital role in Earth's systems. In the oceans, they remain one of the primary producers, organisms that convert sunlight and carbon dioxide into organic matter. They're responsible for a significant fraction of the oxygen we breathe, not just the oxygen produced billions of years ago, but the oxygen we're breathing right now, today. Every breath you take contains oxygen molecules that were likely produced by bacteria in the ocean just months or years ago. Cyanobacteria are also important in many terrestrial environments. They form crusts on soils in arid regions, helping to prevent erosion and fix nitrogen from the atmosphere. They live in symbiotic relationships with plants, particularly with certain ferns and the unusual plant genus Gunnera, providing their hosts with a source of fixed nitrogen in exchange for shelter and nutrients. They even form partnerships with fungi to create lichens, those remarkable organisms that can grow on bare rock and slowly break it down into soil. The story of cyanobacteria and the Great Oxidation Event illustrates a fundamental truth about bacteria. They are not just passive inhabitants of Earth. They are active shapers of the environment. They don't just respond to their surroundings. They change them, profoundly and permanently. And they've been doing this for nearly 4 billion years. Let's talk numbers again, because the numbers tell a story that's hard to grasp any other way. We mentioned earlier that Candidatus pelagibacter ubique might be the most abundant organism on Earth. Let's explore that a bit more, because the story of this bacterium illustrates something important about why bacteria rule the world. Pelagibacter was only discovered in 2002, remarkably recently in the history of microbiology. Before modern genetic techniques, it was effectively invisible to scientists. Traditional methods of studying bacteria involve growing them in laboratory cultures. But Pelagibacter is extremely difficult to culture. It lives in nutrient-poor ocean water and has evolved to be extraordinarily efficient, with one of the smallest genomes of any free-living organism. It has stripped away almost everything unnecessary, becoming a streamlined machine for survival in the open ocean. Once scientists developed ways to detect bacteria directly in environmental samples using genetic techniques, they discovered Pelagibacter everywhere. It's found throughout the world's oceans, from the surface down to moderate depths, in both warm tropical waters and cold polar seas. And there are just so many of them. Current estimates suggest there may be 2 times 10 to the 28th power individual Pelagibacter cells in the global ocean. That's 200 billion bacterial cells, all of this one species. To put this in human terms, if every human on Earth had a pet Pelagibacter bacterium for every atom in their body, we'd still have only accounted for a tiny fraction of the Pelagibacter population. If you could gather all the Pelagibacter bacteria together, they would weigh more than all the elephants, all the blue whales, all the fish, and possibly all the animals combined that have ever lived on Earth. But Pelagibacter is just one species among thousands, perhaps millions, of bacterial species in the ocean. The total bacterial population of the ocean is estimated at about 10 to the 29th power cells. That's 1,000 billion bacterial cells swimming in our seas. They form the base of the ocean's food web. They recycle nutrients, they produce oxygen, they sequester carbon. The ocean's bacteria are, in many ways, the engine that drives marine ecosystems. And it's not just the ocean. Soil is even more densely populated with bacterial life. While we don't have exact numbers for the total bacterial population of all the world's soils, we can make estimates based on what we know about bacterial density in healthy soil. A single gram of soil can contain between 100 million and 1 billion bacterial cells, depending on the soil type, moisture content, and many other factors. If we take a conservative middle estimate of 500 million cells per gram and estimate the total mass of soil in just the top meter of Earth's land surface, we end up with numbers that are staggering. Earth's land surface covers about 150 million square kilometers. The top meter of this soil has a mass of approximately 150 quadrillion kilograms, or 1.5 times 10 to the 17 kilograms. If even a small fraction of this is home to bacteria at the densities we observe in actively growing soil, we're talking about bacterial populations that dwarf even the ocean population. Then there are bacteria living in more extreme environments. Deep beneath the Earth's surface, in cracks and pores in rocks, bacteria have been found living miles below the ground. Some of these deep subsurface bacteria grow incredibly slowly, perhaps dividing only once every 1,000 years, or even less frequently. But they're there, and there are a lot of them. Scientists estimate that the deep subsurface biosphere may contain more bacterial biomass than all the soil and ocean bacteria combined. Bacteria also live in the atmosphere, floating on dust particles and water droplets. They've been found in clouds, where they may actually play a role in weather by serving as nuclei for ice crystals. They live in hot springs and geysers, in acidic drainage from mines, in nuclear waste pools, in the Dead Sea, in the dry valleys of Antarctica, and in the deepest parts of the ocean. Bacteria have colonized essentially every environment on Earth, no matter how extreme, that contains even minimal amounts of water and energy. But perhaps the most startling numbers come from a place much closer to home, your own body. The human body is home to approximately 30 trillion human cells. That's already a large number, hard to fully comprehend, but the number of bacterial cells living in and on your body is even larger, estimated at about 39 trillion cells. You are, in a very literal sense, more bacterial than human, at least in terms of cell count. Most of these bacteria live in your gut, particularly in your large intestine or colon. The colon is one of the most densely populated microbial habitats known, with bacterial cell densities reaching up to 100 billion cells per gram of content. Your gut bacteria collectively weigh about 2 kilograms, roughly the same as your brain. They represent a complex ecosystem, a mini biosphere living inside you, with thousands of different species coexisting, competing, cooperating, and shaping your health in ways we're only beginning to understand. But we're getting ahead of ourselves. We'll come back to the human microbiome shortly. For now, the key point is this: bacteria vastly outnumber all other forms of life on Earth. They exist in greater numbers, greater diversity, and greater total biomass than any other group of organisms. And these numbers aren't just interesting trivia. They reflect a fundamental truth. Bacteria are the foundation of life on Earth. They are the base of the pyramid on which everything else rests. Now let's turn our attention to one of the most intimate relationships we have with bacteria, the ones living inside us. Right now, as you listen to these words, your body is home to trillions of bacterial cells. They live on your skin, in your mouth, in your nose and throat, and most abundantly in your digestive system. These bacteria are so integral to your health and well-being that scientists increasingly think of them not as separate organisms living in us, but as part of us, as an organ in their own right. This collection of microorganisms living in and on you is called your microbiome, and it's so important that the National Institutes of Health launched the Human Microbiome Project to study it in detail. What they and other researchers have discovered is nothing short of revolutionary. Your microbiome isn't just along for the ride. It's actively involved in nearly every aspect of your health, from digestion to immunity to mental health and beyond. Let's start with the gut, where most of your microbiome lives. Your digestive system is home to approximately 39 trillion bacterial cells, representing thousands of different species. This community begins to form from the moment you're born. During birth, babies pick up bacteria from their mother's birth canal, starting the process of colonization. Babies born by caesarean section have a different initial microbiome composition, acquiring their first bacteria from their mother's skin and the hospital environment instead. The composition of your gut microbiome continues to develop and change throughout early childhood, influenced by diet, whether you're breastfed or formula-fed, antibiotic use, and many other factors. By the time you're about 3 years old, your gut microbiome has stabilized into a pattern that will remain relatively consistent for the rest of your life, although it can still be influenced by major changes in diet, antibiotic use, illness, and other factors. So, what exactly are all these bacteria doing in your gut? Quite a lot, as it turns out. First and foremost, they help you digest your food. Humans actually have a relatively limited digestive capacity on our own. We can break down proteins into amino acids. We can digest simple sugars, and we can break down many fats. But there are many components of our diet that we simply can't digest without help. Take dietary fiber, for example. Fiber is incredibly important for health. It helps regulate digestion. It's associated with lower risks of heart disease and diabetes, and it feeds the beneficial bacteria in your gut. But here's the thing. Humans don't actually have the enzymes necessary to break down most types of fiber. We eat it. It travels through our stomach and small intestine largely unchanged. And then it reaches our colon, where our bacteria get to work. The bacteria in your colon have hundreds of enzymes that you don't have. Enzymes capable of breaking down complex carbohydrates like cellulose, hemicellulose, and pectin. They ferment these fibers, breaking them down, and, as a byproduct, producing short-chain fatty acids like butyrate, propionate, and acetate. These short-chain fatty acids are incredibly important. They provide energy to the cells lining your colon, they help regulate your immune system, they influence your metabolism, and they may even affect your brain function. In fact, it's been estimated that these bacterial fermentation products provide about 10% of your daily caloric needs. Your bacteria are literally feeding you, extracting energy and nutrients from food that you couldn't digest on your own. Without your gut bacteria, you would get substantially less nutrition from your food, and you might develop deficiencies in certain vitamins. Speaking of vitamins, that's another crucial role that gut bacteria play. Certain bacteria in your gut can synthesize vitamins that your body needs, but can't make on its own. These include vitamin K, which is essential for blood clotting, and several B vitamins, including B12, folate, and biotin. People who take long courses of strong antibiotics, which can disrupt the gut microbiome, sometimes develop vitamin deficiencies for this very reason. Their bacteria have been wiped out, and suddenly they're not producing these essential nutrients. But the influence of gut bacteria goes far beyond digestion and nutrition. Your gut bacteria are intimately involved in training and regulating your immune system. This might seem counterintuitive. After all, aren't bacteria the things that your immune system is supposed to fight off? Yes and no. Your immune system has to walk a fine line. It needs to be ready to fight off dangerous pathogens, but it also needs to tolerate the trillions of harmless or beneficial bacteria that live in your gut. Your gut bacteria help train your immune system to know the difference. They interact constantly with immune cells in the lining of your intestines, sending chemical signals that help calibrate the immune response. Without this interaction, your immune system might not develop properly. Studies in germ-free mice raised in completely sterile conditions, without any bacteria, have shown that these animals have severely underdeveloped immune systems. Their lymph nodes are smaller, they have fewer antibody-producing cells, and they're much more susceptible to infection when exposed to pathogens. The relationship between gut bacteria and immunity goes even deeper. Your gut bacteria compete with potential pathogens for space and resources, a phenomenon called colonization resistance. When your gut is full of healthy bacteria, there's simply no room for harmful bacteria to get established. The beneficial bacteria are using up the available nutrients, occupying the attachment sites on your intestinal wall, and even producing substances that inhibit the growth of competitors. It's like having a garden so full of desirable plants that weeds can't get a foothold. This is one reason why taking antibiotics can sometimes lead to problems. Antibiotics don't just kill harmful bacteria, they kill your beneficial gut bacteria too. This disruption can allow opportunistic pathogens to move in and cause infections. Clostridium difficile, a bacterium that can cause severe diarrhea and intestinal inflammation, is a classic example. C. Difficile infections typically occur in people who have recently taken antibiotics, precisely because the antibiotics have disrupted their normal gut bacteria and allowed C. difficile to flourish. But perhaps the most surprising discoveries about the gut microbiome involve its effects on the brain and behavior. Scientists have found that there's extensive communication between the gut and the brain, a phenomenon they call the gut-brain axis. This communication goes both ways. Your brain can influence your gut through the nervous system and hormones, which is why stress and emotions can affect digestion. But remarkably, your gut, and specifically your gut bacteria, can send signals back to your brain. Some gut bacteria produce neurotransmitters, the same chemicals that neurons use to communicate. They produce serotonin, dopamine, and GABA, among others. While it's not clear how much of these bacterial neurotransmitters actually reach the brain, there's growing evidence that they can influence brain function and behavior. Studies in mice have shown that altering the gut microbiome can change behavior, affecting things like anxiety and depression. In humans, research has found associations between gut microbiome composition and various neurological and psychiatric conditions, including depression, anxiety, autism spectrum disorders, and even neurodegenerative diseases like Parkinson's disease. It's important to note that most of this research is still in early stages, and we don't fully understand the mechanisms involved. But it's clear that the relationship between our gut bacteria and our brains is much more significant than anyone imagined even a decade ago. The gut microbiome also influences metabolism and weight. Studies have shown that people who are obese tend to have different gut microbiome compositions compared to people who are lean. Even more remarkably, experiments in mice have shown that you can transfer the obesity-associated microbiome from one mouse to another. When researchers transfer gut bacteria from obese mice into thin, germ-free mice, the thin mice gain weight, even though they're eating the same diet as before. This suggests that the gut microbiome itself, independent of diet or genetics, can influence metabolism and body weight. The mechanisms behind this are complex and not fully understood, but it seems that certain bacteria are more efficient at extracting energy from food, and they may also influence hormones that regulate appetite and fat storage. This doesn't mean that gut bacteria are the sole cause of obesity. Diet and genetics clearly play major roles, but it does suggest that the microbiome is another important factor in the complex web of influences on body weight. Given all these important roles, it's not surprising that disruptions to the gut microbiome have been linked to a wide range of health problems. Besides obesity and mental health issues, gut microbiome imbalances have been associated with inflammatory bowel diseases like Crohn's disease and ulcerative colitis, irritable bowel syndrome, allergies and asthma, autoimmune diseases, and even cardiovascular disease. In each case, the relationship is complex, and it's often unclear whether the altered microbiome is a cause, an effect, or simply associated with the disease. But the pattern is clear. A healthy, diverse gut microbiome is associated with better health overall. This has led to great interest in manipulating the gut microbiome to improve health. Probiotics, supplements containing live beneficial bacteria, have become incredibly popular. Prebiotics, dietary components that feed beneficial bacteria are also widely marketed. And in extreme cases, there's even a medical procedure called fecal microbiota transplantation, where the gut bacteria from a healthy donor are transferred to a patient whose microbiome has been disrupted. This procedure has proven remarkably effective for treating recurrent Clostridium difficile infections, and it's being studied for other conditions as well. But our relationship with bacteria extends far beyond our own bodies. Bacteria are essential to the functioning of every ecosystem on Earth, playing crucial roles in cycling nutrients and maintaining the conditions necessary for life. Let's explore how bacteria, despite their microscopic size, are absolutely essential to keeping our planet habitable. Every living thing needs certain elements to survive. Carbon, nitrogen, phosphorus, sulfur, and many others are the building blocks of life, the raw materials from which proteins, DNA, and all other biological molecules are made. But these elements don't stay in one place. They cycle through the environment, moving from the atmosphere to the soil, to living organisms and back again. And at almost every step of these biogeochemical cycles, bacteria are involved. Often, they're not just involved, they're absolutely essential. Without bacteria, these cycles would grind to a halt, and life as we know it would be impossible. Let's focus on one of the most important of these cycles, the nitrogen cycle. Nitrogen is essential for life. It's a key component of amino acids, which make up proteins, and of nucleotides, which make up DNA and RNA. Every living cell needs nitrogen. The ironic thing is that nitrogen is incredibly abundant. About 78% of the air you're breathing right now is nitrogen gas, N2. But that atmospheric nitrogen is in a form that most organisms, including plants and animals, can't use. It's locked up in a very strong triple bond between two nitrogen atoms, and breaking that bond requires an enormous amount of energy. This is where bacteria come in. Certain bacteria, called nitrogen-fixing bacteria, have evolved the ability to break that strong bond and convert atmospheric nitrogen into ammonia, a form that plants can use. This process, called nitrogen fixation, is absolutely essential to life on Earth. Without it, the nitrogen in the atmosphere would be unavailable to living things, and life would be limited to whatever nitrogen happened to be in the soil or water in usable forms. That nitrogen would gradually get used up, and eventually there wouldn't be enough to support much life at all. Nitrogen-fixing bacteria come in two main types. Some are free-living, existing independently in the soil or water. Species like Azotobacter live in the soil and fix nitrogen that nearby plants can use. In the ocean, cyanobacteria fix enormous amounts of nitrogen, making it available to the marine food web. These free-living nitrogen fixers contribute significantly to the global nitrogen budget, fixing tens of millions of tons of nitrogen every year. But the most efficient nitrogen fixers are bacteria that form symbiotic relationships with certain plants, particularly legumes like peas, beans, clover, and alfalfa. These bacteria, primarily species of Rhizobium and related genera, live inside special structures called root nodules on the plant's roots. Inside these nodules, the bacteria fix nitrogen, converting atmospheric N2 into ammonia, and share it with the plant. In return, the plant provides the bacteria with sugars from photosynthesis, giving them the energy they need to power the nitrogen fixation process. This symbiosis is incredibly important. Legumes with their bacterial partners can fix enough nitrogen to meet all of their own needs, and even enrich the soil for other plants growing nearby. This is why farmers often plant legumes in rotation with other crops. The legumes, with help from their bacterial partners, actually add nitrogen to the soil, reducing or eliminating the need for nitrogen fertilizers. Globally, biological nitrogen fixation by bacteria adds about 100 to 200 million tons of nitrogen to the soil each year. But the nitrogen cycle doesn't stop with fixation. Once nitrogen is in the soil in the form of ammonia, other bacteria take over. Nitrifying bacteria convert ammonia into nitrite, and then other bacteria convert nitrite into nitrate. This process is called nitrification, and it's important because nitrate is often the form of nitrogen that plants can absorb most efficiently. These nitrifying bacteria are mostly aerobic, meaning they need oxygen, and they're common in well-aerated soils. Plants absorb nitrogen from the soil in the form of ammonium or nitrate, and use it to build proteins, DNA, and other nitrogen-containing compounds. When animals eat plants, or when other animals eat those animals, the nitrogen moves up the food chain. Eventually, when organisms die, or when they produce waste, that organic nitrogen needs to be recycled back into forms that plants can use again. This is where decomposer bacteria come in. These bacteria, along with fungi and other microorganisms, break down dead organic matter, from fallen leaves to dead animals to waste products. In the process, they release the nitrogen back into the soil as ammonia, a process called ammonification. This ammonia can then be used directly by plants, or converted to nitrate by nitrifying bacteria, completing the cycle. But there's one more critical part of the nitrogen cycle, and it's driven by yet another group of bacteria. In waterlogged soils and sediments, where oxygen is scarce or absent, certain bacteria, called denitrifying bacteria, convert nitrate back into nitrogen gas, which escapes into the atmosphere. This process, called denitrification, completes the nitrogen cycle by returning nitrogen to the atmosphere where it started. At first glance, denitrification might seem counterproductive. After all, if bacteria are working hard to convert atmospheric nitrogen into forms that plants can use, why would other bacteria convert it back into atmospheric nitrogen? But denitrification serves an important function. It prevents the accumulation of excess nitrate in the environment. Too much nitrate in water can cause algal blooms and create dead zones in aquatic ecosystems. Denitrification helps regulate the amount of reactive nitrogen in the environment, maintaining a balance. The nitrogen cycle illustrates a profound truth. The functioning of Earth's ecosystems depends utterly on bacteria. Without nitrogen-fixing bacteria, the nitrogen in the atmosphere would be unavailable, and plants would quickly use up the limited nitrogen in the soil. Without nitrifying bacteria, plants would have difficulty absorbing nitrogen efficiently. Without decomposer bacteria, nitrogen would be locked up in dead organic matter instead of being recycled. And without denitrifying bacteria, excess nitrogen would accumulate and cause environmental problems. Bacteria are the invisible workers that keep the nitrogen cycle, and therefore life itself, running. And nitrogen is just one cycle. Bacteria play similar essential roles in the carbon cycle, helping to break down organic matter and release carbon dioxide back into the atmosphere. They're involved in the sulfur cycle, transforming sulfur compounds in the soil and oceans. They're part of the phosphorus cycle, helping to break down rocks and make phosphorus available to plants. In the oceans, bacteria are key players in the marine carbon cycle, helping to transport carbon from the surface waters to the deep ocean, a process that significantly influences Earth's climate. Without bacteria, the planet would be unrecognizable. Dead organic matter would pile up, never decomposing. The atmosphere would have a completely different composition. Plants would struggle to get the nutrients they need. The oceans would be chemically very different. In short, the Earth would not be able to support life as we know it. Bacteria are not just inhabitants of Earth, they are maintainers of the conditions that allow all other life to exist. Now, let's venture into some of the most extreme environments on our planet. Places where you might think nothing could survive. Boiling hot springs, where water bubbles up at temperatures high enough to cook you in seconds. Frozen wastes, where temperatures rarely rise above zero. The crushing darkness of the deep ocean, where the pressure would instantly flatten any unprotected human. Acidic pools, where the pH is low enough to dissolve metal. Incredibly salty lakes, where the salt concentration is so high that no fish can survive. In each of these environments, life does exist, and almost always, that life is bacterial. These bacteria, called extremophiles, have evolved remarkable adaptations that allow them to not just survive, but thrive in conditions that would quickly kill any other organism. Their existence teaches us profound lessons about the resilience and adaptability of life, and about the remarkable diversity of bacterial strategies for survival. Let's start with heat. Most organisms can't survive temperatures much above 40 or 50 degrees Celsius. At these temperatures, proteins start to denature, losing their shape and therefore their function. DNA can break apart. Cell membranes can become too fluid. For most life, high temperatures are deadly, but thermophilic bacteria, which means heat-loving bacteria, can live at temperatures up to around 80 degrees Celsius. Hyperthermophilic archaea, close relatives of bacteria, can survive at temperatures above 100 degrees Celsius, even up to 120 degrees in some cases. These heat-loving microbes are found in hot springs, geysers, and hydrothermal vents on the ocean floor. One of the most famous is Thermus aquaticus, discovered in a hot spring in Yellowstone National Park. This bacterium thrives at about 70 degrees Celsius. It might seem like just an interesting curiosity, an odd organism living in an unusual place. But T. aquaticus has had an enormous impact on human society. Scientists isolated an enzyme from this bacterium, a DNA polymerase that can withstand high temperatures. This enzyme, called Taq polymerase, became the key to a revolutionary technique called polymerase chain reaction, or PCR. PCR is used to make many copies of specific DNA sequences, and it requires repeated heating and cooling cycles. Most enzymes would be destroyed by this heating, but Taq polymerase, evolved to work at high temperatures, survives just fine. This has made PCR one of the most important tools in modern biology. It's used in medical diagnostics, forensics, genetic research, and even COVID-19 testing. A bacterium from a hot spring in Yellowstone has helped transform science and medicine. But how do thermophiles survive such high temperatures? They've evolved several adaptations. Their proteins are stabilized by additional chemical bonds and by changes in their amino acid composition that make them more resistant to heat. Their cell membranes contain special lipids that remain stable at high temperatures. Their DNA is protected by special proteins and by an increased ratio of guanine-cytosine base pairs, which form 3 hydrogen bonds instead of 2, and are therefore more stable. At the opposite extreme are psychrophiles, cold-loving bacteria that thrive at temperatures near or below freezing. These organisms are found in polar regions, in alpine environments, in the deep ocean, and in permafrost. Some can remain active at temperatures as low as -15 degrees Celsius. Like thermophiles, psychrophiles have evolved special adaptations. Their enzymes are flexible enough to work at low temperatures. Their cell membranes contain unsaturated fatty acids that remain fluid even when cold. Some produce antifreeze proteins that prevent ice crystals from forming inside their cells. Then there are the halophiles, salt-loving bacteria and archaea that live in extremely salty environments. Some can survive in salt concentrations approaching saturation, where the water is so full of salt that no more will dissolve. The Dead Sea, salt flats, and commercial salt evaporation ponds are home to these remarkable organisms. Many halophiles are pigmented red or purple, giving salt flats their distinctive colours. Living in high salt conditions presents unique challenges. Water naturally flows from areas of low salt concentration to high salt concentration, a process called osmosis. In a very salty environment, water would tend to flow out of a cell, dehydrating it. Halophiles solve this problem in various ways. Some accumulate high concentrations of salt inside their cells to balance the external salt concentration. Others produce organic molecules called compatible solutes that balance the osmotic pressure without interfering with cellular functions. Acidophiles live in highly acidic environments with pH values as low as 0, about as acidic as battery acid. These organisms are found in acid mine drainage, volcanic hot springs, and other naturally acidic environments. They keep the inside of their cells at a nearly neutral pH while living in acid that would corrode metal. Conversely, alkalophiles thrive at pH values above 9 or 10, living in soda lakes and other alkaline environments. Perhaps most remarkable are the organisms living deep in the ocean at hydrothermal vents. These vents form where seawater seeps into cracks in the ocean floor, gets heated by magma beneath, and then shoots back up in superheated plumes. The water can reach temperatures of 350 degrees Celsius or more. It doesn't boil because of the enormous pressure at these depths, but it's hot enough to melt lead. Around these vents live entire ecosystems based not on sunlight, but on bacteria that can harvest energy from the chemicals in the vent fluid, particularly hydrogen sulfide. These chemosynthetic bacteria form the base of a food web that includes giant tube worms, clams, crabs, and many other creatures, all living in complete darkness miles below the ocean surface. The bacteria live inside the tissues of the tube worms, protected from the extreme conditions, and provide nutrients to their hosts in exchange for shelter and access to the chemicals they need. The discovery of these hydrothermal vent ecosystems in the 1970s was revolutionary. It proved that life didn't need sunlight to thrive, that whole ecosystems could be powered by chemical energy. It also raised tantalizing possibilities about life on other worlds. If life can thrive in the dark, hot, chemically rich environment of a hydrothermal vent, could similar life exist in the subsurface oceans of Europa or Enceladus, moons of Jupiter and Saturn, where liquid water may exist beneath thick ice shells? Barophiles, also called piezophiles, love high pressure. They live in the deep ocean, where pressures can reach over 1,000 atmospheres, over 1,000 times the air pressure at sea level. These bacteria have adapted their cellular machinery to function under crushing pressure. Their proteins are stabilized by unique structural features, and their membranes remain functional even when compressed. Some bacteria are polyextremophiles, meaning they can tolerate multiple extreme conditions simultaneously. Thermoacidophiles, for example, live in hot, acidic environments like those found in volcanic areas. These organisms face the double challenge of high temperature and high acidity, and they've evolved remarkable solutions to both. The study of extremophiles isn't just fascinating science for its own sake. These organisms have practical applications. Enzymes from thermophiles are used in industrial processes that require high temperatures. Compounds from halophiles are used as preservatives. DNA polymerases from thermophiles are essential for biotechnology. And extremophiles give us clues about the limits of life, information that's invaluable as we search for life on other planets. But perhaps the most profound lesson from extremophiles is this: life is far more adaptable and resilient than we ever imagined. Wherever there's liquid water and a source of energy, however minimal, life finds a way. The habitable zone of Earth isn't limited to the comfortable middle ground of temperature and chemistry that we mammals enjoy. It extends into almost every niche, every crack and crevice, every pond and puddle, no matter how extreme. And in almost all of these extreme environments, it's bacteria leading the way, pioneering survival strategies that push the boundaries of what we thought was possible. Have you ever wondered whether bacteria are aware of each other? It might seem like a strange question. Bacteria are single-celled organisms with no brain, no nervous system, no obvious way to sense their environment beyond simple chemical detection. And yet, bacteria are far from the isolated, independent entities we once thought them to be. They communicate, they cooperate, they coordinate their activities in ways that seem almost impossibly sophisticated for such simple organisms. This phenomenon is called quorum sensing, and it's one of the most fascinating discoveries in microbiology of the past few decades. The basic idea is deceptively simple. Bacteria release chemical signals into their environment. As the bacterial population grows, the concentration of these signals increases. When the concentration reaches a certain threshold, it triggers changes in gene expression in the bacteria, coordinating their behavior as a group. Quorum sensing was first discovered in a bioluminescent marine bacterium called Vibrio fischeri. This bacterium lives in symbiosis with certain species of squid and fish, residing in special light organs. The bacteria produce light through a chemical reaction. But here's the interesting thing. Individual bacteria in dilute solution don't glow. They only start producing light when they reach high cell density. Scientists discovered that V. fischeri produces a small molecule called an autoinducer. This molecule diffuses freely across the bacterial cell membrane. When there are only a few bacteria, the autoinducer diffuses away into the environment and its concentration stays low. But when bacteria are packed together at high density, like they are inside a squid's light organ, the autoinducer accumulates. When it reaches a high enough concentration, it binds to proteins inside the bacteria, triggering the expression of genes responsible for light production. Suddenly, all the bacteria in the population start glowing together. This is quorum sensing, bacteria releasing chemical signals, detecting the concentration of those signals, and changing their behavior when the population reaches a certain density. It's a clever system that allows bacteria to act collectively, to do things as a group that wouldn't make sense for an individual cell. Think about it from the bacteria's perspective. Producing light requires energy and resources. For a single bacterium floating alone in the ocean, producing light would be wasteful. The light wouldn't be bright enough to be noticed by anything, and the bacterium would just be wasting resources. But when many bacteria produce light together, the combined glow is bright enough to be visible and useful. Quorum sensing ensures that bacteria only invest in light production when there are enough of them for it to matter. But quorum sensing isn't limited to controlling bioluminescence. Scientists have discovered that bacteria use quorum sensing to control a wide range of behaviors, and many of these have serious implications for human health and disease. One of the most important is biofilm formation. Biofilms are communities of bacteria that stick to surfaces and encase themselves in a protective matrix of polymers. Think of the slime that builds up on rocks in a stream, or the plaque on your teeth. That's a biofilm. Bacteria in biofilms are much more resistant to antibiotics and to the immune system than free-floating bacteria. Many chronic infections involve biofilms, and they're a major problem in hospitals, where they can grow on catheters, implants, and other medical devices. Many bacteria use quorum sensing to regulate when they form biofilms. When bacterial numbers are low, the cells remain free-floating, able to move and find new environments. But as their population grows and quorum sensing signals accumulate, the bacteria change their behavior. They start producing the sticky polymers that form the biofilm matrix. They attach to surfaces, and they build the three-dimensional structure of the biofilm. This coordination ensures that biofilms form only when there are enough bacteria to make the investment worthwhile. Quorum sensing also controls virulence, the ability of pathogenic bacteria to cause disease. Many disease-causing bacteria don't immediately start attacking their host when they first arrive. Instead, they wait until their population has grown large enough. They use quorum sensing to coordinate the expression of virulence factors, toxins, and enzymes that help them invade tissues, evade the immune system, and cause disease. Pseudomonas aeruginosa, a bacterium that can cause serious infections, especially in people with weakened immune systems or cystic fibrosis, is a classic example. P. aeruginosa uses multiple quorum sensing systems to control the production of toxins, enzymes, and biofilms. When the bacterial population is small, these virulence factors aren't expressed. The bacteria are relatively harmless, quietly growing and reproducing. But once the population reaches a certain size, quorum sensing kicks in, virulence factors are expressed, and the bacteria become aggressive, causing tissue damage and inflammation. This makes evolutionary sense. If bacteria express their virulence factors immediately upon infecting a host, they might provoke a strong immune response before they had time to establish a secure foothold. By waiting until their numbers are larger, they increase their chances of overwhelming the host's defenses. It's a strategic delay, a coordinated attack that's much more effective than an uncoordinated one. The discovery that bacteria use quorum sensing to control virulence has led to an exciting new approach to fighting infections: quorum sensing inhibition. Instead of trying to kill bacteria with antibiotics, which can lead to resistance, why not interfere with their communication? If you could block quorum sensing, bacteria would never coordinate their attack. They might remain in the host, but they wouldn't express their virulence factors, and they wouldn't cause disease. The immune system could then clear them at leisure. Several research groups are working on developing quorum-sensing inhibitors. Some of these are molecules that mimic autoinducers but don't activate the response, essentially jamming the bacterial communication channel. Others are enzymes that break down autoinducers, preventing them from accumulating to the threshold concentration. Some organisms have evolved their own quorum-sensing inhibitors as a defense against pathogenic bacteria, and scientists are studying these natural products for potential medical applications. Quorum sensing isn't just used for communication within a species. Some bacteria can detect and respond to signals from other bacterial species, allowing for interspecies communication. There's even a molecule called AI2 for autoinducer 2 that's produced by many different species of bacteria and appears to function as a universal signal for microbial communication. This allows bacteria to sense not just the density of their own species, but the overall bacterial population density in their environment. And the communication goes even further. Some bacteria can detect and respond to signals from eukaryotic organisms, including plants and animals. This interkingdom communication can influence everything from plant-bacteria symbioses to the virulence of pathogens in human hosts. It's a complex web of chemical signals, with bacteria constantly monitoring their environment, sensing the presence of other organisms, and adjusting their behavior accordingly. The phenomenon of quorum sensing has fundamentally changed how we think about bacteria. We used to view them as simple, independent cells, each one going about its business without regard for its neighbours. But quorum sensing reveals that bacteria have sophisticated mechanisms for social behaviour. They communicate, they cooperate, they make collective decisions. In a very real sense, a population of bacteria acts like a multicellular organism, with individual cells coordinating their activities for the benefit of the group. This has profound implications. It means that understanding bacterial behaviour requires looking not just at individual cells, but at populations. It means that bacteria are capable of much more complex behaviours than we realised. And it means that we may have new ways to combat bacterial infections, not by killing bacteria, but by disrupting their ability to communicate and coordinate. Now let's turn to a topic that's increasingly urgent in modern medicine: bacterial resistance to antibiotics. This is a story about evolution in action, about the remarkable adaptability of bacteria, and about how our use and misuse of antibiotics has created one of the most serious health threats of our time. It's also a powerful demonstration of why bacteria, with their rapid reproduction and incredible genetic flexibility, rule the world. When penicillin was first introduced in the 1940s, it seemed like a miracle. Infections that had been death sentences were suddenly curable. Soldiers who would have died from infected wounds survived. Diseases like pneumonia and sepsis became treatable. The discovery and widespread use of antibiotics is one of the greatest achievements of modern medicine, saving millions of lives. But even as penicillin was being rolled out, scientists were already observing something troubling. Some bacteria weren't killed by penicillin, they were resistant. Alexander Fleming himself, who discovered penicillin, warned about the potential for resistance to develop if antibiotics were used improperly. Sadly, his warnings went largely unheeded. The problem is rooted in basic evolutionary principles. antibiotics create a strong selective pressure on bacterial populations. When you expose bacteria to an antibiotic, the vast majority of cells will be killed. But if even a few cells have some resistance, perhaps because of a random mutation, those cells will survive and reproduce. Since bacteria can divide every 20 minutes under ideal conditions, a single resistant cell can quickly give rise to millions of descendants. Over time, the population shifts from mostly susceptible bacteria to mostly resistant ones. This is evolution, happening right before our eyes, on timescales we can observe directly. It's not a slow process that takes millions of years. It can happen in a matter of days or weeks. And it's been happening constantly since we started using antibiotics. Bacteria have evolved resistance to every antibiotic we've ever developed. Some bacteria are now resistant to multiple antibiotics, earning them the terrifying nickname of superbugs. Methicillin-resistant Staphylococcus aureus, or MRSA, is resistant to most beta-lactam antibiotics. Multi-drug-resistant tuberculosis is resistant to the two most powerful anti-TB drugs. Carbapenem-resistant Enterobacteriaceae, or CRE, are resistant to carbapenems, antibiotics that were considered last resort treatments. Some strains of bacteria are now resistant to all known antibiotics, making infections with these organisms essentially untreatable. The mechanisms by which bacteria become resistant are diverse and sophisticated. Some bacteria develop enzymes that break down antibiotics before the drugs can act. Beta-lactamase enzymes, for example, cleave the beta-lactam ring in penicillin and related antibiotics, rendering them inactive. There are now hundreds of different beta-lactamase enzymes, each capable of breaking down different antibiotics, and bacteria continue to evolve new ones. Other bacteria develop mutations that alter the target of the antibiotic. Most antibiotics work by binding to specific bacterial proteins or cellular structures. If the shape of that target changes, the antibiotic may no longer be able to bind effectively. For example, some bacteria have developed mutations in their ribosomal RNA that prevent certain antibiotics from binding, making the bacteria resistant to those drugs. Some bacteria develop pumps that actively push antibiotics out of the cell. These efflux pumps use energy to export various toxic substances, including many antibiotics. By pumping the antibiotic out as fast as it enters, the bacteria keep the internal concentration low enough that the drug can't do its job. Bacteria with these pumps can be resistant to multiple different antibiotics at once, a phenomenon called multi-drug resistance. Still, other bacteria alter their cell membrane to prevent antibiotics from entering in the first place. By changing the structure or composition of their outer membrane, they can block certain antibiotics from getting inside the cell where they would normally act. But perhaps the most concerning aspect of antibiotic resistance is the ability of bacteria to share resistance genes with each other through horizontal gene transfer. We touched on this earlier when discussing bacterial evolution more generally, but it's particularly important in the context of antibiotic resistance. Bacteria can transfer genes through several mechanisms. In conjugation, two bacteria temporarily connect and pass DNA between them, often in the form of small circular DNA molecules called plasmids. These plasmids can carry genes for antibiotic resistance, and they can be transferred even between bacteria of different species. In transformation, bacteria take up DNA from their environment, often from dead bacteria. In transduction, viruses that infect bacteria can pick up bacterial genes, including resistance genes, and transfer them to other bacteria. This horizontal gene transfer means that antibiotic resistance can spread much faster than it could through simple reproduction and inheritance. A bacterium that has evolved resistance to an antibiotic can share that resistance with its neighbours, even neighbours of different species, within hours. This is why we see antibiotic resistance spreading so rapidly through bacterial communities, including communities in hospitals and in the human microbiome. The situation is made worse by the widespread use and misuse of antibiotics. In many countries, antibiotics are overused in human medicine, prescribed for viral infections where they're completely ineffective, or used without proper testing to determine which antibiotic would be most appropriate for the specific infection. In agriculture, antibiotics are used extensively in livestock, not just to treat sick animals, but as growth promoters and for disease prevention in healthy animals. This exposes huge populations of bacteria to sublethal doses of antibiotics, creating ideal conditions for resistance to develop. When antibiotic-resistant bacteria develop in livestock, they can be transferred to humans through meat or through environmental contamination. When antibiotics are overused in humans, resistant bacteria can spread from person to person. And once resistance genes enter the global pool of bacterial DNA, they can persist and spread indefinitely, being shared and reshared among bacterial populations worldwide. The consequences of widespread antibiotic resistance are serious. Infections that were easily treatable are becoming difficult or impossible to cure. Procedures that rely on antibiotics to prevent infection, like surgery and chemotherapy, are becoming riskier. The World Health Organization has declared antibiotic resistance one of the top 10 global public health threats. Some have warned of a return to a pre-antibiotic era, where simple infections could once again be deadly. But the story of antibiotic resistance isn't just a tale of doom. It's also a powerful demonstration of bacterial adaptability and evolutionary speed. Bacteria have shown, again and again, that they can evolve solutions to almost any challenge we throw at them. The very traits that make bacteria so successful, their rapid reproduction, their huge populations, their ability to share genes, these same traits make them incredibly difficult to control. This is part of why bacteria rule the world. They're not just ancient, not just abundant, not just essential to ecosystems. They're also incredibly adaptable. They can evolve at speeds that large organisms simply can't match. When we try to control them with antibiotics, they evolve resistance. When we try to exclude them from sterile environments, they find ways in. When conditions change, they adapt. The antibiotic resistance crisis is driving scientists to look for new approaches to fighting bacterial infections. Some are searching for new antibiotics, particularly ones that bacteria haven't been exposed to before. Others are looking for ways to make existing antibiotics more effective, like combining them with drugs that block efflux pumps or inhibit resistance enzymes. Still, others are exploring alternatives to antibiotics altogether, like bacteriophages, viruses that specifically infect and kill bacteria. There's also growing interest in precision medicine approaches that would allow doctors to quickly identify the specific bacteria causing an infection and choose the most effective antibiotic, rather than using broad-spectrum antibiotics that kill many different types of bacteria, and thus create stronger selective pressure for resistance. And there's work on developing vaccines against bacterial pathogens, preventing infections rather than trying to treat them after they've already established themselves. But perhaps, most importantly, there's a growing recognition that we need to use antibiotics more carefully. We need to prescribe them only when they're truly necessary. We need to complete courses of treatment to ensure bacteria are fully eliminated rather than just suppressed. And we need to drastically reduce the use of antibiotics in agriculture. These are not easy changes to make. They require changes in medical practice, agricultural practices, and public policy. But they're necessary if we want to preserve the effectiveness of antibiotics for future generations. The story of antibiotic resistance is a sobering reminder of the power of evolution and of the remarkable adaptability of bacteria. But it also underscores something more fundamental. Bacteria aren't just passive inhabitants of our world. They're active participants, constantly evolving, constantly adapting, constantly finding new ways to survive. They've been at this for nearly 4 billion years, and they're very, very good at it. Let's shift now to a slightly more enchanting aspect of bacterial biology: their ability to produce light. Bioluminescence, the production of light by living organisms, is one of nature's most magical phenomena. We're all familiar with fireflies flashing on summer evenings, but bioluminescence is actually much more common in the ocean, where it's estimated that up to 76% of pelagic organisms can produce light. And at the base of this glowing ocean world are bacteria. Bioluminescent bacteria, about 30 species that we know of, are found throughout the world's oceans. Most belong to the genera Vibrio and Photobacterium. These bacteria produce light through a chemical reaction involving an enzyme called luciferase, which catalyzes the oxidation of a substrate called luciferin. When luciferase and luciferin react in the presence of oxygen, they produce light, typically in the blue-green part of the spectrum, around 490 nanometers wavelength. This particular wavelength isn't random. Blue-green light penetrates seawater better than any other color, so it makes sense that marine organisms would evolve to produce light in this range. It's the most efficient wavelength for communication and signaling in the ocean environment. But why would bacteria produce light at all? Light production is energetically expensive. It requires resources that could otherwise be used for growth and reproduction. For bioluminescence to evolve and persist, it must provide some benefit that outweighs this cost. Scientists have proposed several hypotheses about the function of bacterial bioluminescence, and the truth likely involves multiple factors. One well-established function is that bioluminescent bacteria form symbiotic relationships with marine animals, particularly fish and squid. These animals have special light organs that house dense populations of bioluminescent bacteria. The bacteria receive a safe home with plenty of nutrients, while the animal gets a source of light that it can use for various purposes. The Hawaiian bobtail squid, a small nocturnal squid about the size of your thumb, is a classic example. These squid have a special light organ in their mantle cavity that's colonized by the bioluminescent bacterium Vibrio fischeri. At night, when the squid is active, it hunts for small shrimp in shallow waters. Moonlight and starlight penetrating the water would normally create a silhouette of the squid, making it visible to predators lurking below. But the squid uses its bioluminescent bacteria in a clever way: counter-illumination. The squid adjusts the amount of light produced by the bacteria to match the intensity of moonlight and starlight penetrating from above. This eliminates its silhouette, making it essentially invisible from below. During the day, the squid buries itself in the sand and expels about 90% of its bacterial population, then allows the remaining bacteria to regrow overnight. Many fish have similar light organs housing bioluminescent bacteria. Some use the light for counter-illumination, like the squid. Others use it for communication, for attracting mates, or for luring prey. The flashlight fish has light organs beneath its eyes containing photobacterium bacteria. The fish can cover and uncover these organs with special membranes, effectively creating a blinking light that it uses to communicate with other flashlight fish and to confuse predators. But not all bioluminescent bacteria live in symbiosis. Many are free-living in the ocean or live on the surface of marine organisms or on marine detritus. For these bacteria, the function of bioluminescence is less clear, but recent research suggests it may help bacteria get eaten. This might sound counterintuitive. Getting eaten doesn't seem like a good survival strategy, but hear me out. When bioluminescent bacteria colonize a particle of organic matter, like a bit of dead plankton or a fecal pellet, they make that particle glow. This glowing particle is much more visible to zooplankton and small fish, which are attracted to the light and eat the particle. The bacteria survive passage through the animal's gut and are deposited elsewhere in the animal's feces, along with a rich supply of nutrients from the animal's digestive process. From the bacteria's perspective, getting eaten is actually a form of dispersal. They hitchhike in the guts of fish and zooplankton, being transported to new locations where they can colonize new particles and new environments. The light they produce is like a beacon saying, "Come eat me." And it works because animals are attracted to light in the dark ocean. But there's another layer to this story. Remember quorum sensing, the bacterial communication system we discussed earlier? It turns out that bioluminescence in bacteria is controlled by quorum sensing. Bacteria only produce light when they reach high population density. This makes sense because light from a single bacterium would be far too dim to be detected. Only when many bacteria produce light together does the combined glow become visible. In Vibrio fischeri, the quorum sensing system that controls bioluminescence was one of the first bacterial communication systems to be discovered and characterized. Individual V. fischeri cells floating freely in the ocean don't glow, but when they colonize a squid's light organ and reach high density, they start producing light en masse. The quorum sensing system ensures that bacteria only invest resources in light production when there are enough of them for the light to be useful. This connection between quorum sensing and bioluminescence has been incredibly important for science. Researchers use bioluminescent bacteria as model organisms for studying bacterial communication. The light production provides an easy-to-measure output. You can literally see when quorum sensing is activated. This has made it possible to study the details of how bacterial communication works, leading to insights that apply to many other bacterial species and systems, including pathogenic bacteria that use quorum sensing to control virulence. Beyond their ecological role, bioluminescent bacteria have proven valuable for biotechnology and scientific research. The genes for bacterial luciferase can be inserted into other organisms, allowing scientists to create glowing bacteria, plants, and even animals. These glowing organisms can be used as biosensors, lighting up in the presence of specific chemicals or environmental conditions. They're used to study gene expression, to track the spread of infections in real time, and to screen for potential new drugs. The discovery and characterization of bacterial luciferase has even contributed to the development of some medical technologies. Luciferase-based assays are used in research and drug development. The principle of bioluminescence imaging, where scientists use bioluminescent markers to track biological processes, was pioneered using bacterial systems. But perhaps the most profound thing about bacterial bioluminescence is simply the wonder of it. In the dark depths of the ocean, bacteria are producing light through chemistry, creating their own tiny lamps through biochemical reactions. Some of the most beautiful sights in the ocean, the sparkle of bioluminescent dinoflagellates in breaking waves, the glow of jellyfish, the blinking lights of deep-sea fish, all of these have their roots in the biochemistry that bacteria evolved hundreds of millions of years ago. As we near the end of our journey tonight, let's step back and consider what we've learned about why bacteria rule the world. We've seen that they are by far the most abundant organisms on Earth, outnumbering all other life forms by staggering margins. We've seen that they've been here longer than any other form of life, surviving and thriving for nearly 4 billion years through ice ages, asteroid impacts, and countless other changes. We've seen that they created the very atmosphere we breathe and continue to maintain the conditions necessary for all other life to exist. We've explored how bacteria are essential partners in our own bodies, with trillions of bacterial cells living in our gut and on our skin, helping us digest food, produce vitamins, train our immune systems, and even influencing our brains and behavior. We've seen how bacteria drive the biogeochemical cycles that keep nutrients flowing through ecosystems, how they can survive in the most extreme environments on Earth, and how they communicate and cooperate in sophisticated ways. We've confronted the sobering reality of antibiotic resistance, a powerful demonstration of bacterial adaptability and evolutionary speed, and we've marveled at some of their more enchanting abilities, like the production of light in the dark ocean depths. In every one of these areas, bacteria demonstrate capabilities that put larger, more complex organisms to shame. They're more numerous, more diverse, more adaptable, and more essential than any other form of life on Earth. They've shaped the planet and continue to shape it. They've made it possible for complex life, including us, to exist. And they continue to play absolutely crucial roles in maintaining the habitability of our world. But perhaps most remarkably, they do all of this while remaining largely invisible to us. We go about our daily lives completely unaware of the trillions upon trillions of bacteria around us and inside us. We don't see the bacterial populations in the soil beneath our feet, recycling nutrients and helping plants grow. We don't see the bacteria in the ocean, producing oxygen and driving food webs. We don't see our own gut bacteria working tirelessly to help us digest our food and protect us from pathogens. This invisibility is part of why it's taken us so long to appreciate how thoroughly bacteria dominate Earth. We naturally focus on what we can see, the plants and animals at our own scale. But the microscopic world of bacteria is just as real, just as important, indeed more important than the visible world we're familiar with. Understanding this, truly grasping the extent to which bacteria rule the world, should change how we see our place in nature. We're not the dominant species we thought we were. We're relative newcomers, living in a world that bacteria built and continue to maintain. We depend on bacteria, absolutely. They do not depend on us. We exist because bacteria made the Earth habitable, and we continue to exist only because bacteria continue to perform services that we couldn't live without. This isn't meant to be humbling in a negative sense. Rather, it's an invitation to appreciate the true complexity and wonder of the natural world. Life on Earth isn't just about the organisms we can see. It's about vast networks of microscopic organisms performing countless biochemical reactions, cycling elements through ecosystems, evolving at incredible speeds, adapting to every available niche, and collectively shaping the conditions that allow all other life to persist. And there's still so much we don't know. Despite over 300 years of studying bacteria, since Leeuwenhoek first observed them through his handmade microscopes, we're still discovering new bacterial species, new bacterial capabilities, and new roles that bacteria play in ecosystems. The vast majority of bacterial species have never been cultured in laboratories. Many have never even been formally described. There are entire groups of bacteria whose existence we only know from their DNA sequences in environmental samples. As our tools for studying bacteria become more sophisticated, as we develop better methods for sequencing bacterial genomes and studying bacterial ecology, we're discovering that the bacterial world is even more complex, more diverse, and more important than we realized. Every new study seems to reveal new ways that bacteria influence the world, from their roles in cloud formation to their effects on climate, from their participation in global element cycles to their influence on animal behavior. The story of bacteria is far from complete. They continue to evolve, to adapt, to spread to new environments, and to interact with other organisms in ways we're only beginning to understand. They will be here long after humanity is gone, just as they were here long before we arrived. They've survived every mass extinction event in Earth's history, and they'll almost certainly survive whatever the future holds. So, yes, bacteria rule the world. They always have, and they almost certainly always will. And far from being a disturbing thought, this should fill us with wonder. We live in a world shaped and maintained by invisible organisms, a world of chemical conversations and ancient partnerships, of extreme survival and exquisite adaptation. We live in a bacterial world, and it's more amazing than we ever imagined. Thank you for joining me tonight on Bedtime Biology. If these explorations into the quiet wonders of nature help you unwind and spark your curiosity, please consider liking this video and subscribing to the channel. And I'd love to hear from you in the comments. Let me know where you're checking in from tonight, and what aspect of bacterial life you found most fascinating. Until next time, sleep well, and remember, you're never alone. You carry an entire microbial world with you wherever you go. Sweet dreams.